To start the process for our data analytics, we're going to download the file. So the first one we're going to do is Descriptive Statistics. So we're going to take this file here, download it please, and note that it's a CSV file. That's what we got to upload. Don't change this to an Excel file. This is what we're going to be using. Next, we're going to go to Jupyter at jupyter.org. I'm going to try it in our browser, and we're going to use Jupyter Lab. You can use Jupyter Notebook as well. There's just a couple extra buttons you got to click to be able to upload your files. So I found this one's much easier. So if I click on here, we'll see that I've got an extensive array already of files that I've used. But essentially, go to notebooks. You'll see that with this here, I can click here. I can get data and notebooks. I found that if I put my data sets in here that it has a problem trying to read it. So I stay in notebooks. I keep my notebooks in here and my data is in here as well. So what you're going to want to do then is go to the upload files and upload specifically the lab data. Make sure it's the file that you want. And I've already got it, so I don't need to do that. Once you've got it in there, you can take a look and see that that's what you have. So I can double click on this and I can see the data set now. It should be relatively clean. There's empty spaces here, but it's essentially the same data set that I've had before. And now we can continue to actually work on Jupyter Notebook with this. All right, so let's start off by creating a new data set, or sorry, a new lab notebook. So I'm going to go here to the launcher. I can either do it here, I can do the plus sign. I'm going to go here and add Python, and we are in. Some things to note here, just watch there's a kernel over in this thing that is active. That's what we want to happen. So first thing we can do is do a markdown code to actually kind of describe what we're going to do. This is a descriptive analysis of lab data or whatever it is that we need. Remember, from the markdown language, we can put in a variety of different things to make the title bigger or smaller, but whatever, as long as it actually gives us some information. So first thing we need to do, we're going to import pandas because that's our main thing that we're going to be using. And the next one we're going to use then is matplotlib. One other important thing I want to tell you here is that as I go through, notice how the words are not highlighted here. So that's something is going on right now, and we will check and see what it is. So and we'll get another file, another one. So there is something going on here. So I'm going to try to run this file and see if it'll actually run that code. And as you can see, really nothing's happening because it doesn't recognize that these things are active right now. So a couple of options I can do. Typically, you can go and restart the kernel, or you can run and restart kernel and run all cells. I want to restart. And hopefully what that'll do is it'll get that back and active again. The other thing too is that I don't actually have this saved. So I'm going to save this as a descriptive analysis and see if that will help solve the problem as well. So we'll go back up, redo this, and see if we can get it to go. Eventually I had to shut down the browser, restart it again, and I was able to get it to finally get a kernel going. And then when I finally ran it, I have a spelling error right here. So I had this and it told me that I am trying to put in pylot instead of pyplot. So now that I've got all that in there, I'm going to try restarting the kernel again. And this is it. So essentially, I'm importing pandas, I'm importing matplot library or lib, and so we're going to create a data frame. So we're going to call in this data set here, the Chem 10046 lab 0, and we're going to do this describe function on it. So this is going to create the data frame that we're looking for. It's going to have all the different headings and whatnot that we need, and we're going to then describe it. So just actually give us some information or data. Again, my kernel doesn't seem to want to run. We'll see if we can get it to go again. If not, I'm going to have to shut down the browser once again. So I find that this is the biggest pain that I've got when dealing with this. So here is the output then. Pretty simple, very straightforward. So the data set that we had, now we've got all the titles here that are exactly that was in our data set. So we can call that up just to take a look. So there's the titles of all the data sets right there. This is our information that we've got. And so now The describe function then tells us how many variables are in each one. So for year, there's 242, there's 241 in mass salicylic. A couple of things to note. The first is that not all columns are actually in here. So there's one here called lab day, and it has strings. It does not have numerical value. And the descriptive analysis only works on numerical data, so you'll find that that column is missing. So for future though, when we want to do group by, we'd like to see all of these columns up here. So we're probably going to need a df columns in here. So let's just try that out and see if it'll work for us. So on our data frame, we want to see what the columns are. So just type that in and I'm going to run that one. And so we'll see there's lab day. So I find that this function is very good for actually figuring out lab day. So if I want to do some further analysis here, I can highlight the exact name. I can do it here as well if I need it for a variable later on, which is what we're going to do. So use these types of things to your advantage to actually get the proper title instead of typing it. When you type it by hand like I did here, you can actually have some errors associated with it. So out of this again, so we've got the count, we've got the mean. Some of the information is useful, some of it's not. So the mean for the year over from 2017 to 2023, this actually isn't a very useful bit of information. So we can actually remove this if we wanted to, and only specifically get the means and standard deviations for things that we want. So let's go to just the mass of salicylic acid. This is what you would weigh out in the very first lab. Every student has to weigh out this stuff. And so for the past 7 years or so, this is the average that students have weighed out, 4.999247. If we were to go with the significant figures here, we go to 4 decimal places. The standard deviation then, 0.2561, and the minimum value, one student weighed out 2.9 grams, and the max value, someone weighed out 7 grams. So we've got pretty high max there and min. And so now we deal with what are called the quartiles. So this 50% one here, this is our median. So this is where half the data is below this value and half the data is above this value. Similarly with the 25%, 25% is below this value, 75% is above this value. And with the 75%, so 75% of the data is below this value and 25% of this data is above this value. So between 75 and the max, you're going to find 25% of the data. Between 25% and the min, you're going to find 25% of the data in there. So that's what this descriptive statistics tells us. It's really straightforward, very, very simple. Let's say we just want to get the information for one of the columns specifically. So we're on the data frame, we're going to call in one of the columns. So we'll just say mass salicylic. I'm going to copy everything, including the quotes, Control C, and then Control V. I'm going to end the square brackets, and then I'm going to describe like this. And if we run that, we'll see that we get the information just specifically for this one. If we want more than one column, I'm going to Control C, Control V, but I can actually highlight more than one, so including the comma here. So I'm going to highlight this whole set, do the Control C again, put that into its place here. But if I have more than one column here, I'm going to need to put in double brackets. I don't know why, I just do. If I do that and run it, you should see 2 columns worth of data now. So, and it makes it look a little bit fancier here when it does it as well. All right, so that's one way you can do it. So I could have called in very specifically which ones I wanted and get all the information for it. So very, very simply. So this information is great, but the one thing that we're missing is this is, say, the mean for the entire data set from 2023 back to 2017. What if we wanted to get the average per year? So we're going to need to use a new function called the group by function. So now I'm going to create a new variable. So let's say it's called group data or grouped data. And I'm going to have that equal to the data frame, and I'm going to use this group by function. So there we go, that's the new one. So I'm now going to put in specifically what column I want to group by. So I'm going to highlight this one, Control C. Again, I do this because I find it's easier for me to make mistakes, to not make mistakes. So there we go. So we've now created a new variable called group data. I've got the data frame, and I'm going to group by specifically the year. And so we're going to create some statistics. So I'm going to just create a new variable. I don't have to. I can do it on specifically this group data. I could have put dot describe right after here as well, but I'm going to just say statistics equals the grouped data. I'm going to make sure I spell this right, or try to spell it right. And specifically, I want it for the mass salicylic. So let's go up here and get our title for mass salicylic. Put that in. So this is going to group year by year, and it's going to do the statistics, the describe function that we're about to do, specifically on the mass of salicylic acid column. So let's put in describe, and that's our function. So the hope is here that we're grouping by year, so we should see an output for year by year, and specifically the mass of salicylic acid, we want the descriptive statistics for that. So if we run this, let's hope for the best. And I've spelled something wrong. Let's just see group data. Oh, I put an extra capital in there. Let's try this again. And now my kernel doesn't want to work. It is funny that every time I want to correct something, change it, the kernel disappears on me. There it is right there. So usually I have to shut down my browser and get it started again. So I'll try one more time here and see if I can get it to go. Nope. Oh, it worked. I didn't have faith in it. All right. So we haven't got our data here yet because I think I need to print the statistics because I got a variable print statistics. I didn't actually tell it to print this out because it's a variable. All I did was say it's the statistics equals all of this information. I didn't actually tell it to print it out. So let's hope that I spelled that right. Run that one. And so now you'll see year by year, here's how many data points are in 2017. This is the mean specifically for 2017, the standard deviation. So the file changed slightly, or at least it transposed a little differently. But there's the data that we wanted right there. So year after year after year, like that. So it's a pretty neat little tool to be able to easily and effectively change everything. Again, if I wanted to group the statistics by mass salicylic and say massive acetic anhydride, I could put that in there and I get more data spread out through this as well. So let's say I want to do the same thing, but I want to do it by lab day. So I'm just going to copy this cell. I could actually do copy cell as well. But instead instead of by year, I want to do it by lab day. So I'm going to go back up here. I'm going to specifically copy this, Control C, put it into here, Control V. I'm going to describe the mass of salicylic, but I want it by lab day. And of course, I need to print this. So I'm going to print, or try to spell it right. Why don't I just copy this? There we go. I already know it works. All right, so let's see if we can run this cell. And there we go. So here, the first one we did group by year, and so there's each year spitting out the information. Now we're doing it by lab day, and we're able to compare everything. So where this comes in really handy is when we're going to do t-tests and the ANOVA, because the information we want to compare, say, lab day by lab day or year by year to see some sort of trend or just see if everything is consistent or not. So a very, very nice way of actually controlling our data and taking a look. I can also do this based off of lab day and year. So if I were to take this again, put it into this cell, like this. And so now here I want to do both year and lab day. So I'm going to, in the square brackets, I'm going to put year. I'm just going to type it in because hopefully I can get it correct. And I want a comma here and lab day. And we will hope that this works. Right now it doesn't look promising because the rest of this stuff is all in red right now. And I'm afraid if I run this, I'm going to have an error. So let's try and see what happens. Oh, there we go. So my year is done differently than that. Okay, so this looks better. The print shows up in green, describe is in blue, and this comma is in black. So all of that is much better looking now. And there we go. So when we do it now, we've grouped it by year and by lab day. So in 2017, here are the lab groups. So very specifically in 2017, here are the lab groups, and we can see how many data points there are for each one of these um, uh, specific sections. And we got to go across the column here. This little thing tells us that we've got it. The rest of the column is over here. But the mean for 2017 for the very first lab day there was 4.9 and then 5.0. So again, very, very nice way of actually getting the data out very, very quickly and easily. As long as you spell everything correctly, you're good to go. So we got a lot of data coming out here and we've got it organized the way we want. Let's say we just want the mean and we don't want all the rest of this information. What I can do, I'm going to just copy this again, bring it down here, paste it. But what I'm going to do is I'm going to change this. This statistic is for everything. I just want the mean. So I'm just going to call it mean. And we'll say it's for salicylic. Hopefully I spelled that right. The group data is going to be based on mass salicylic, and instead of the entire describe function, I'm just going to do the mean. And I could do standard deviation, and I could do just the max and min if I wanted to as well. And I'm going to print, but I'm going to print this new variable that I just created. Oops. So I'm going to put a bracket at the end of this. All right, so what we've done is we've grouped the data by or create a new variable based on the data frame and group by from year and the lab day. I created a new variable called salicylic. I didn't need to. I could have put mean right on the end of this and the group data. I could have printed that and it would have been a group by and then the dot mean would be here. I did a second one. It's not very Pythonic. You want to make everything as quick as possible, but it's easier for me to visualize and to group everything together like this. So if we run this, hopefully, we see here now that we've only got the mean data out and that all the rest of the stuff here, all the other columns aren't in here. This becomes useful now because I can now plot this data. So in the next video, that's what we're going to do.